Hi. Start over.
Yeah.
Hi. I can't do that with my eyes. All right, now I'm just going to say, hi, I'm Allie,
My name's Haley.
and today we're going to talk about soil microbes.
Woohoo! What's a microbe?
Microbes are tiny, and that's how we define them. So microbes are micro. It means that we can't see them with our naked eyes. We need to use a microscope in order to see them. So it's really funny classification because we're just saying that they are this thing because humans can't see them.
So they're like little itty bitty creatures that we have to have a microscope to see, and that's what makes them a microbe?
Yes, we have to have a microscope to see them, but if lots of them grow, then we can see what a bunch of them look like at once.
Well, what does just one look like? Can we get a picture? So what kind of microbes live in soil?
Lots of different microbes, and some of the categories of microbes you may find are bacteria,
Okay.
yeasts, viruses, and algae.
Okay. Algae lives in soil?
Yeah.
All right.
So where have you heard of algae from?
Well, lakes.
Mhm.
Sometimes they turn green, like green algae.
Yeah. So they can also live in soils.
But they're also microbes.
Yeah.
Okay.
But we see them because there's so many of them that they grow to be big. And another really interesting type of a microbe is nematodes. Nematodes are tiny worms, and sometimes they're micro, and then sometimes they're macro. So sometimes we need a microscope to see them, but sometimes they're big enough just to see.
So can we grow them?
We can grow microbes.
Ooh, I want to grow some microbes.
Absolutely. So all we need to do to grow microbes are to give them a habitat that they like. And the cool thing about microbes is that they like to live in all different kinds of places. And in fact, we can make them one habitat, and they will go and live in different parts of it based on what they can find to live, what they can live on there. So what their favorite food is, or what kind of thing that they breathe.
So what do I need to do? I need to get some microbes. Where do I get microbes?
Well, you can get soil microbes from a great way to find a bunch of interesting soil microbes is to go to a stream and get some mud from the bed, stick it in a jar.
All right, I'm going to do it.
All
Stay
right.
tuned. We're at the beginning of our Winogradsky experiment at the middle of Coney. So this is the upper part of the middle of Coney, right down from Bear Creek Reservoir. These are the shoals, and specifically where we're standing is a Native American fishing weir. Recently there was a lot of floodwaters that came through here, and so if you look over this way, there's a good deal of deposition. That's where we were planning to get some of our mud for this experiment, but because of all of the sand, we're gonna get it from the bank over here. And so we're choosing an area where we're thinking we're gonna get some good diversity of microbes. And so I'm digging into this side of the bank there, and putting it in a bucket that already has a little bit of water from the middle of Coney, so that we'll have that for our experiment. And I guess just to kind of get a sense of the texture here, so you can see it's got a good deal of clay and sand. And you know, it's got that earthy smell, but there's also just a wee bit of a sulfur smell. Might as well pull out some of these roots, organic matter. We're not gonna want that for our experiment. You can see this is probably more recent deposition of the sand, but we're gonna go for this more clayey mud for the soil. So that's our location of the soil.
So now we're going to talk about the things that you'll need to assemble your Winogradsky columns. First, for the container for the column itself, I have two plastic quart containers because we're going to make two columns. And you could use a plastic jar, you could use a 2-liter soda bottle with the top cut off. You could use a plastic water bottle, but it might be a little harder to get the mud in. So you might need to use a funnel and might have to thin your mud down. So that's why I used something with a wide mouth. You can use a glass jar, but don't tightly lid it if you use a glass jar because gas is going to build up and you don't want the jars to explode. So you could put some tin foil or plastic wrap with a rubber band on top if you're using a glass jar. Next we have our mud from the middle of Coney, and we've got a nice little trowel to move our mud with. We have water from the middle of Coney, and we have some nutrient inputs. So our nutrient inputs include a egg yolk, which will be a source of carbon and also sulfur. We have, for more carbon, some cut up paper. You could use newspaper. I used some grocery bag handles and grocery bags. And then I also have the eggshell from the egg smashed up. This gives us some calcium. And then the last things that you'll need are tinfoil, because we're going to set up an experiment where we cover one of our Winogradsky columns totally with tinfoil. You don't have to do an experiment where you have two different, like a control and an experimental. You can just make one column if you want to just observe. And also we have some tape and marking pens so we can label our experiments. All right, now for the fun part. Let's play with some mud. So you're just going to use your trowel to move the mud from one container to your Winogradsky column. You don't want to include like really big leaves or sticks or anything like that. So if you see those in there, this is actually pretty good. I was going to do an example, but I can't find any. If you see some in there, you can just throw them in your compost. So I'm going to fill it up a little more, and then we're going to mix in those nutrient sources. So this is the food for our microbes. The reason I cut up the paper so small is just so that we can get it nice and evenly distributed throughout the mud. And you can mess around adding some other inputs if you want. If you have, for instance, like an iron source, like a rusty nail lying around, you could stick that in there. Any other like mineral sources you can think of. If you have garden lime, you could add that. It's really kind of up to you, but the basics are just adding a carbon source and a source of sulfur. All right, that's looking pretty good. I'm going to top it off with just a little more mud. And then all we need to do is add our water. So top it off, maybe an inch of water. And you want to make sure that you leave some headspace, both because, as I said, like gas is going to be produced, so you can leave a little space for that gas to happen, but also you might see some neat things growing at this interface between the water and the air. So you want to make sure that it's not all the way up top where you can't see it when you cover it up. Okay, so now we have both our columns made. We have our mud, our water. We covered them with plastic wrap and a rubber band, and then they are labeled with the source of our mud, our nutrient inputs, and then this one is labeled with a light bulb because this one will be exposed to the light. And then this one is our experimental. I mean, really either could be experimental and either could be control, but we're going to call this is on the control, exposed to the light. And this one, the experimental, we're going to see what happens when the column is not exposed to the light. And by covering it with tinfoil, we will accomplish this. Good cut. All right, it was a little awkward, but I got this one covered in tinfoil. And now we're going to put them in their home where they're going to live for the next month and a half, 2 months. And so we want this to be exposed to the light. So basically put it in a windowsill. You could put it in a protected area on your porch, somewhere where you can get light, like where you would put a houseplant. Okay, so here we have our Winogradsky columns a couple of days after we made them. We put them in the sun, and some of the clay particles have settled out of the water, so it's gotten clearer, which is great because then we'll be able to see what's going on in there. And we're just gonna keep checking on the columns every few days, noting what they look like, and probably the first indication that microbes are growing in there will be bubbles. So keep an eye out for any little bubbles traveling through the water. That means your microbes are growing, and then over the next few weeks, you'll be able to observe some different changes indicating microbial life.